Welcome back to the fourth video of our online classes in pharmaceutical microbiology. In the previous classes, we have seen introduction to microbiology. After that, we have seen what prokaryotes and eukaryotes are. And then in the third video, we have seen the classification of bacteria. We have first seen the structure of a bacteria, the cell membrane and cell components, right? And after that, we have seen the classification of bacteria based on morphology, anatomy, and so on. In today's class, we'll be studying about the bacterial cultures, okay? And what are the requirements for a bacteria to grow, okay? And so today's class objectives will be these. So first thing, we need to know what culture media is, okay? And after that, what are the requirements of culture media? As we are human beings, we eat food, and food that we eat needs vitamins, minerals, and all, right? Similarly, microorganisms require all this stuff in order to grow. They need some nutritional medium to survive and some other nutritional medium to grow further into various generations. And then we have to know what are the different types of culture media present and which are commonly used, and what are the composition or concentration of various culture medias. So beginning with what culture media is. Basically, culture media is an artificial environment. What do you mean by artificial environment? Suppose if we eat a fruit or a vegetable, we get vitamins and minerals, isn't it? That is by natural source, and we are taking it. And that vitamins are being given to us, isn't it? Similarly, in case of microorganisms, they don't have anything, they don't have mouth to eat, right? We have mouth, so we are eating. We are able to administer things. Whereas microorganisms is not like that. They absorb by osmosis or diffusion process, right? In the same way, microorganism needs some nutritional medium to get. And how do they get the nutritional medium? We need to provide certain nutrients like nutrients or vitamins in order for the microorganism to grow. So we are making the nutrient medium, right? So it is an artificial medium, right? Isn't it? Whatever we create will be the artificial, which is from the natural is natural, and whatever we create, that will be artificial, right? So the culture media, we can define it as an artificial environment that supplies all the required nutrients for the microorganism to survive and show its growth. You can see the image here. These are various microorganisms, various different types of culture media. So we'll be studying what are those culture medias in the coming slides. One more thing to remember is there are certain microorganisms which does not survive or does not produce even in artificial environment. That is the culture media. What could be the reason? These are the parasites within our body. Let us take Mycobacterium tuberculosis, that is tuberculosis. Those bacteria, when they get into our body, they start reproducing. So they need a host in order to reproduce and survive. Such bacteria are called obligate bacteria or obligate parasites. These parasites need a host cell in order to survive. If there is no host cell, they cannot survive. So you can read this point. Most of the microorganisms can be cultured in the culture medium except obligate parasites. And what are they? Those which cannot survive in the absence of host cell. And the examples can be Mycobacterium leprae. I said Mycobacterium tuberculosis before. So let me correct. This Mycobacterium leprae, which causes leprosy, and Trypanosoma pallidum, which needs host cell in order to survive. Okay. Moving on to the requirements of culture media. As I have said in the beginning of the class, we need certain nutrients in order to survive, like proteins, vitamins, minerals, isn't it? Similarly, microorganisms need certain require microorganism need certain things in order to survive. It can be chemical requirements and physical requirements. Let us see what chemical requirements are. Chemical requirements are the carbon, okay, and nitrogen, sulfur, phosphorus, trace elements such as manganese, copper, all these, and organic growth factors, and vitamins and oxygen. Oxygen is required because they can be either aerobic bacteria or anaerobic bacteria, right? And coming to physical requirements, temperature, pH, hydrostatic pressure, osmotic pressure. This temperature we have discussed in the classification. Even based on temperature, the bacteria are classified, right? Thermophiles, psychrophiles, and all. Okay, and based on pH also we have classified certain bacteria, and based on no hydrostatic pressure, but there's certain pressure required in order to for the bacteria to grow, and osmotic pressure. This was the requirements for the culture media, basic requirements for all culture media. Okay, and moving on to the classification of culture media. So in the beginning we have studied what was culture media, and the next slide we have seen what was the requirement of a culture media. Okay, the chemical requirements and the physical requirements. Chemical will be carbon, nitrogen, trace elements, right, vitamins, oxygen. Whereas physical parameters could be temperature, pH, salt condition, salt concentration, right? And what was the other one? It was hydrostatic pressure and osmotic pressure. Osmotic pressure you can you can also consider as salt concentration. So beginning with the constituency of culture media. Basically, culture media can be classified based on three things, and that will be composition, consistency, and purpose. Basically, the food can be of two types or three types. It could be solid, semi-solid, or liquid. In our case, consider the rice that we eat or the chapati that we eat. They are solid in nature. Whereas if we go for upma, upma is semi-solid, right? And when we go for fruit juices, they are completely liquid. So solid, semi-solid, and liquid, right? Similarly, the culture media can be of three types based on the constituency. So it could be solid media, semi-solid media, liquid media. How is solid media formed? In order for the solid media to form, there should be uh, certain polymer present, not polymer, certain substance present which will be able to solidify the nutrient medium. In case of rice, we have starch within the rice, and for that reason, it has a specific shape. Whereas in chapati, it has a flour, it also has starch, and we get a particular shape, right? Let it be fruits, they have a particular shape. But solid media, how do they get shape? How do they get rigidity? They get rigidity because of a gelling agents. Gelling agent can be gelatin or agar. Basically, in culture media, agar will basically used. Okay, in solid media contains agar, that is 1.5 to 2%. Other solidifying agents. It has physical structure as it is a solid, so it has physical structure, and it is used for isolating bacteria. In the culture media, we can see different spots forming. So those spots will be it could be different bacteria or colonies of same bacteria, colonies of different bacteria. Okay, so basically this will be used to segregate or isolate different bacteria. Coming to semi-solid media, semi-solid, that will be uh, like upma I have said you. Similarly, it contains less than 0. Equal to or less than 0.5% of agar. This agar is gelling agent or solidifying agent. So it has a custard like constituency. We have seen custard how it is. It will be semi-solid. If you put a spoon, it easily breaks, right? So similarly, the semi-solid media will be like that of custard. So it is used for cultivating not the plants, it is the microorganisms. But excuse me, it is cultivating microorganisms. Okay, and you can see the last point in semi-solid media, useful for the cultivation of microaerophilic bacteria or for determination of bacterial motility. Suppose it is a semi-solid one, and if the bacteria is present at one end, it can move to the other end. So in this, we can be able to find if the bacteria is moving or not, if it has a mobility or not. Whereas in solidifying bacteria, we will just be able to see the formation of a spot of a bacteria, colony of a bacteria, but we cannot see if the bacteria is moving or not. Moving on to the liquid media, also called as broth. Okay, it contains nutrients but no gelling agents. In solid and semi-solid, gelling agent that was agar was present, whereas in liquid media, agar will be absent. Okay, gelatin or agar, and it is used for production of producing generation. Sorry, it is used for producing generations of organisms, fermentation studies and tests. So as you know, the liquid media It is used for producing a number of generations of the bacteria. In solid media, we are able to isolate bacteria, right? We are able to isolate. In semi-solid, we are able to cultivate them. And similar to semi-solid, the liquid media is also helpful to cultivate bacteria, producing a number of generations of the bacteria. So moving on to the next classification, that is based on composition. The composition can be based on composition can be of two types. One is synthetic and non-synthetic. Synthetic can be chemically defined. Non-synthetic is chemically non-defined or undefined. So what is chemically defined? When I say chemically defined, it means we know what is the chemical compounds present in that solution or present in that medium. So it is basically made from purified ingredients. Whatever the ingredients used to prepare that culture is completely pure. There won't be any contamination. And exact composition is known, as I just said you, chemically defined, right? So exact composition is known and can be simple or complex. Whereas on non-synthetic or chemically undefined, in this we won't be able to find the exact concentration of the compounds added. So contains at least one component partially digest, that will be partially digested proteins. Partially digested proteins can have bacteria, right? They can have bacteria because they are going for digestion, but it's not completely digested. So it can be contaminated, or it can have some other enzymes also. So it can be simple or complex. Simple non-synthetic nutrient medium or culture medium meets nutrient requirements. What it means is the simple culture medium will just be able to support the life of the bacteria. It cannot uh, cause the bacteria to grow in a number of generations. It will just make the bacteria to survive, that's it. Whereas the complex nutrient medium or non-synthetic nutrient medium, it will be able to provide nutrients such that the bacteria can survive, as well as it keeps on reproducing, so that it can produce a number of generations. So support growth of fastidious microorganisms. Moving on to the next classification based on purpose. On what purpose? Is it aerobic bacteria that we are cultivating or anaerobic? Is it gram positive or gram negative? Based on that, that will be our purpose. What is basic purpose, right? It could be some antibiotics that we are isolating, or it could be like we want to check the quality of antibiotic, right? We can also check the assay of antibiotic. At what concentration is it inhibiting the growth of bacteria? So many special purpose media are needed to facilitate recognition, enumeration, that is, um, uh, is, enumeration means it will be resoluting, it will be identifying each and every bacteria present, and isolation of certain types of bacteria. To meet these needs, numerous media are available, okay, as just discussed. Various types of nutrient mediums are available, okay, and they are as follows. First is general purpose media or basic media. Next is enriched media. The next is selective and enriched media. The next is differential or indicator media, and transport media, anaerobic media, assay media. We'll see each and every media in brief now. So general purpose media or the basic media. For human beings, for survival, the basic thing that we need is food, and it can be rice. Rice is the basic food required for human to survive. Similarly, for the bacteria to survive, it needs some basic requirements. Okay, and that basic requirement can be peptone water, nutrient broth, and nutrient agar. So this the petri dish that you see, the round one, is the nutrient agar. Okay, and whereas this one is a peptone water. So basically, basal media are basically simple media that supports most non-fastidious bacteria. Okay, this is a basic nutrient media that supports the life of a bacteria. And what will be including in the basic medium or basal medium could be peptone water, nutrient broth, and nutrient agar. And these media are generally used for primary isolation of microorganisms. So I just said this will just support the bacteria to survive, not grow in numbers. It will be helpful to just survive. When it is able to survive, we can also see different spots forming or different colonies forming, and based on that we can isolate the bacteria. Moving on to the enriched media. What is enriched media? The word itself says we are making the culture media rich. If someone gives me money, I'll be rich, right? I myself won't be rich. If someone gives me money, I'll be rich. If I earn, I'll be rich, right? Similarly, in culture media, if some extra thing is added, that will be called as enriched media. So what are added, we'll see now. Addition of extra nutrients in the form of blood, okay, the form of blood, serum, egg yolk, etc. to basal medium makes them enriched media. So blood, serum, egg yolk are, when they are added to the basic or basal medium, they are called as enriched media. So this, why do we use blood in this? There are certain bacteria which cause hemolysis. Hemolysis means breakage of the cells or RBCs, and RBC is red in color. So when blood is added to the agar solution, okay, when blood is added to the agar solution, we call it as blood culture media or blood agar media. Okay, in this, when we add a microorganism that causes hemolysis, we will be able to see the round spots on the culture media. Okay, and that will help in identifying if the microorganism is causing hemolysis or not. So the best examples can be blood medium or chocolate medium. Blood medium is adding blood to the agar solution, okay, and chocolate medium is when blood is heated, when it is heated with up to 50 or 60 degrees centigrade, or even 40 to 60 degrees centigrade, it turns brown in color. Okay, that blood when heated forms brown in color. That is called chocolate medium. The next is selective and enriched media. What is selective mean? Selective means uh, it needs certain things, okay, certain selective things. If that is absent, bacteria cannot survive, it will definitely die, or it cannot grow further. Okay, let us see. Selective and enrichment media inhibits unwanted commensals contaminating bacteria and helps to recover pathogen from bacteria mixture of bacteria. What does it mean? It means, suppose a culture media is added with microorganism. It can have various types of bacteria. Suppose I take the soil from the ground, add water in that, and I try to culture that one. So soil has n number of bacteria, right? So if I want to isolate only one bacteria, how can I do that? I need to add something which kills all other bacteria except one which I need, right? Similarly, in this differential growth suppression, which means growth of other bacteria will be prevented, and it favors the growth of one type of bacteria. That we'll see now. Differential growth suppression suppresses the growth of some microorganisms, right? I just said it suppresses the growth of some microorganisms while allowing the growth of others. These are agar-based solid medium so that individual colonies may be isolated. So basically, the nutrient medium will be solid, and upon adding selective compounds that kills all other microorganisms except that I need, it will kill all other microorganisms, and what I need will retain there. Okay, that will grow and form different colonies. Based on it, I can isolate it. So what are the various approaches to make the medium selective? And that will be addition of antibiotics, addition of dyes in case of gram positive and gram negative, and addition of chemicals and altering pH, or a combination of these. So addition of antibiotics can help in killing all other microorganisms and growing one organism. And dyes, gram positive or gram negative, we can identify. And chemicals, I've just discussed, it could kill other microorganisms similar to that of antibiotics. And alteration of pH, in the solution that we have taken, it is possible that there are certain microorganisms within that which will die at higher pH and survive at lower pH, or vice versa. Okay, and let us see the examples of selective media. Mannitol salt agar and salt milk agar, used to recover Staphylococcus aureus, and contains 10% of sodium chloride. So mannitol salt agar, it contains mannitol in it, and salt is also mentioned. So 10% of sodium chloride will also be present. What it does is it kills all other bacteria except Staphylococcus aureus. Okay, this bacteria will survive and starts growing. Even if it is dying, it helps in recovering the bacteria and keeps on growing. So potassium telluride medium used to recover diphtheria, Clostridium diphtheria and contains 0.04% of potassium telluride. Okay, this compound presence in 0.04% uh, helps the Clostridium diphtheria to recover if it is going down or dying. The next is McConkeys agar used for Bacteriaceae members, contains bile salt that inhibits most gram-positive bacteria. It inhibits most of the gram-positive bacteria and simultaneously it favors the growth of gram-negative bacteria. And next is pseudosol agar, cetrimide agar. It contains cetrimide, which is an antiseptic agent. Dettol that we use is antiseptic, right? Similarly, cetrimide is another agent that is antiseptic. So this cetrimide helps to recover this Plasmodium aeruginosa. Okay, whereas it kills all other bacteria. Next is crystal violet blood agar used to recover pyrogens containing 0.002% and contains 0.002% of crystal violet. Basically, you might have seen what crystal violet is. It is blue in color, and if you have an aquarium at home, you'll be adding some blue color solution within the tank so that the water doesn't get contaminated. What is that blue color? That is crystal violet, or sorry, that is methylene blue. Similar to that will be crystal violet. Okay, and it's 0.002% of crystal violet present, helps in recovering of Staphylococcus pyrogens. Okay, so Streptococcus pyrogens. And next is Lowenstein-Jensen medium used to recover Mycobacterium tuberculosis and is made selective by incorporating malachite green. Malachite green is another reagent. Okay, when this is added to the medium, it makes the medium selective and helps in recovery of Mycobacterium tuberculosis. And the last is Wilson and Blair's agar for recovering Staphylococcus, sorry, Salmonella typhi. Okay, that causes typhoid. So it helps in recovering Salmonella typhi by selectively adding dye brilliant green. Brilliant green is a dye used to add in the culture medium in order to grow or in order to recover the Salmonella typhi in the culture media. So these were the examples of selective media. There are various other examples as well, but these are the most important. Moving on with the differential or indicator media. What does it mean? The word itself says indicator media. That means it indicates something with, how does it indicate? With basically when we drive a vehicle, we have indicators on the left and on the right, which indicates to which direction we are going to turn, to which direction we are going to turn. If it is left, it will be on the left side. If it is the right, it will be on the right side. Similarly, in this case, this culture media, based on the bacteria present, it shows various colors. Let us see how it is. So different bacteria can be recognized on the basis of their colony color. Okay, called as indicate. Okay, since it is able to differentiate the bacteria based on the colony color, and that color, different color, means it is indicating that particular bacteria is present, and hence it is called as indicator media. You can see the image here. The yellow color one, this is in the same media. Okay, yellow color one is Staphylococcus aureus. Okay, and the right one is Staphylococcus epidermidis. These two are different bacteria. Yellow color is aureus and the pink color is epidermidis. This is in the same culture media, right? This is the same culture media, which is differential indicator media. In that, different color indicates different bacterial colony. So various approaches include how do we make the culture media as indicator media or differential media? That is by adding dyes, okay, incorporation of dyes, metabolic substrates, and etc. So that bacteria utilize them, appear as differently colored colonies. And these media allow the growth of more than one microorganism of interest, but with morphologically distinguishable qualities. Okay, it can be identified by the morphology itself. So the best examples can be mannitol salt agar, okay, that we discussed just in the previous slide. Mannitol agar, This maltol fermentation produces yellow color. Blood agar that we have dealt before. Various kinds of hemolysis, that is alpha, beta, and gamma hemolysis can be found. McConkey's agar that we just saw the last slide. Lactose fermenters, pink color colonies, whereas non-lactose fermenters produce paler colorless colonies. So basically, this is the culture media. Which culture? It can be McConkey agar because it produces pink color colony. Next one is transport media. What is transport media? Suppose if I have something, or if I have prepared something, a dish, I need to transport it from one place to another place. If I have to send it to my relative's house, I'll be packing in a box and then sending it. I'll make sure that the food doesn't get spoiled or it doesn't get spilled out. If the curry is it doesn't get spilled out, right? Similarly, the microorganisms, when they're being transferred from one institution to other institution, right, from a hospital to the lab, or from one organization to other organization, it should be prevented so that it won't spill out and cause diseases, right? So it has to be confined in a particular vessel. How does that vessel? Not all vessels are most protective for microorganisms. It can cause spilling of the bacteria. So there are certain culture media which holds the bacteria for some time, so that until unless the organism is completely transported. So used in case of transporting microorganisms, such media prevent drying of specimen. What does it mean, drying of the specimen? During the transport, if I just let us take banana itself. If I take a banana, put it open for and keep it for 5 to 6 days in the sunlight, what happens? Complete edible part gets dried up and the skin becomes completely dry, and it will be like a paper, right? That means the water content is completely going away. That means it is getting desiccated. The water is going completely away. Same thing, if microorganisms are taken just like that, the desiccation happens. The contents present within the bacterial cell gets destroyed, they get evaporated or they get removed out. As a result, bacteria will die. So in order to prevent that, this type of media helps the bacteria to not desiccate. So they prevent drying of specimen, okay, or desiccation of specimen, maintain the pathogen to commensal ratio and inhibit growth of unwanted bacteria. So basically, it prevents the growth of unwanted bacteria and also maintains the commensalism, or the bacteria which uh, ratio between the pathogen, the disease causing organism, and the organism which is commensal, that doesn't cause any harm to the body. Okay, these two are maintaining a ratio in order to transport the bacteria. And some of these media, stewards and amines, are semi-solid in consistency. Okay, semi-solid media. Addition of charcoal serves to neutralize inhibitory factors. So since it is semi-solid, there can be possibility of hydrolyzing enzymes present in that. So basically, addition of charcoal prevent neutralizing inhibitory factors. And the carry bearer transport medium Venkataraman Ramakrishnan medium are used to transport feces from suspected cholera patients. What happens is, in case of cholera, the patients suffering from cholera caused by Vibrio cholera, their faces are taken, okay, the excretory material of the humans, or the excretory of the humans, that faces are taken and transferred to a lab and checked if uh, if if the cholera is present or if it is absent. And if they want to transfer from one organization or other organization, so they use this type of pair-bed transfer medium or Venkataraman Ramakrishna medium in order to culture it and transport to the other organization. Similarly, SACH buffered glycerol saline is used to transport feces from patients suspected to be suffering from bacillary dysentery, and Pike's medium is used to transport streptococci from throat specimens. So this was the transport media, and next is anaerobic media. Why do we need to discuss about anaerobic media? As you know, certain bacteria are aerobic or anaerobic. They survive in the presence of oxygen or in the absence of oxygen. Studying of the anaerobic media is very important because presence of small concentration of the oxygen also kills the microorganisms. So how is it? How is the preparation of the anaerobic medium is? We'll see now. So it needs special media, right? It shouldn't have any oxygen concentration. So reduce oxidation, reduction potential, and extra nutrients. It should have a low concentration of oxygen and should have extra nutrients in order to support the life of microorganism. So media have to be supplemented with nutrients like amine and vitamin K. Such media may also have to be reduced by physical or chemical means. It means the nutrients has to be added carefully so that oxygen won't be in a higher concentration. So boiling the medium, even by boiling the medium, uh, all the normally when we boil water, the gas bubbles we see that those gas bubbles are the dissolved oxygen and carbon dioxide present in the water. That will be that gases will be removed out when we boil. Similarly, on boiling the culture media, the dissolved oxygen present will be expelled out. So expels any dissolved oxygen, and after boiling, after expelling the dissolved oxygen, it should be sealed with a liquid paraffin, sterile liquid paraffin. Okay, we get a strips of paraffin, liquid paraffin. We get a strips, and that we can just uh, cover it and seal it tightly. Next is Robertson cooked meat, that is RCM medium. It is commonly used for growing Clostridium species and contains 2.5 centimeter column of bullock heart meat. Okay, bullock heart. Bullock, as you know, the cow bullock, that bullock's heart meat, 2.5 centimeter column, and 15 ml of the nutrient broth, and thioglycolate broth, which contains thioglycolate, glucose, cysteine. Cysteine is the amino acid, and yeast extract and casein hydrolysate. This is an enzyme. So this was a composition of Robertson cooked meat. And next one is methylene blue. As I just discussed before, that some solution will be added in the aquarium in order to prevent the growth of bacteria. That is methylene blue, right? So methylene blue or Resazurin. Even if you see the bottle of solution that you add in the aquarium, you will be able to see the name methylene blue. It is an oxidation reduction potential indicator that is incorporated in the medium. Under reduced condition, methylene blue is colorless. Okay, and the assay media. What is assay media? As I mentioned about the finding the concentration of antibiotics at which it can inhibit bacteria, that is the assay. It means it will be helpful to determine the potency of antibiotic or other drug. Let us see this. These media are used for assay of vitamins, amino acids, and antibiotics. Example, antibiotic assay media are used for determining antibiotic potency of the microbial assay by the microbial assay technique. So what basically they do is they find the IC50 value. What is IC50? Inhibitory concentration 50, which means that 50% of the concentration of the drug, to what extent is it inhibiting the growth of microorganisms? That will be found by antibiotic assay method. Okay, and for these different types of medium include media for enumeration of bacteria. Enumeration means isolation or yeah, isolation and identifying the different colonies of the bacteria, and media for characterization of the bacteria, that is the morphology or anatomy, and maintaining the media. So these were the various media that is basically used for the culture of microorganisms. We have been seeing the media, what media it is. Okay, we have seen various names of it. So what is the composition of them? Let us see. Basically, nutrient broth that we have. Nutrient broth contains 50 grams of meat. Example, ox heart. The before we have seen bullock heart meat, right? Here in nutrient broth is 50 grams of meat, that is ox heart, which is minced and mixed with 1 liter of water. Minced means it is completely like it is completely made to very small particle. It is completely made to paste and mixed with 1 liter of water, 1 liter of the water, and 10 grams of peptone, sodium chloride are added. pH is adjusted to 7.3. So this will be the culture media for that is nutrient broth. Nutrient agar. Basically, agar is added. Okay, 2.5% of the agar is added at heated at 37 degrees. That is room temperature, a bit higher than the room temperature. Okay, and it is heated. Okay, sorry, agar is basically a solid at 37 degrees centigrade. That is almost the room temperature. So when it is boiled at 100 degrees centigrade, it forms a liquid. We'll see here it is heated at 100 degrees centigrade to melt the agar and then cooled. Then peptone water, that is 1% of the peptone plus 0.5% sodium chloride. That is a mixture of peptone water. The composition of peptone water is 1% of peptone and 0.5% of sodium chloride solution. That mixture forms peptone water. So in Newton agar, the Newton basically agar is solid at 37 degrees centigrade, and when it is heated at 100 degrees centigrade, it forms a liquid, and to it peptone water is added, which contains 1% of the peptone and 0.5% of the sodium chloride. And what is blood agar? Most commonly used medium. Okay, this is the most commonly used medium, especially to find hemolytic bacteria, and it contains 5 to 10% of defibrinated sheep or horse blood. So defibrinated means removal of the fibrogen from the blood. Okay, fibrogen is a protein that helps in clotting of a blood. So if that fibrogen is removed, the blood will not clot. Okay, and 5 to 10% of defibrinated sheep or horse blood is added to the melted agar at 45 to 56 degrees centigrade. Blood acts as an enrichment material, hence it is included in enriched culture media and also as an indicator. Certain bacteria, when grown in blood agar, produce hemolysis around the colonies. Based on that hemolysis causing, we can identify if the bacteria is hemolytic bacteria or not. And next is chocolate agar or heated blood agar. So it is prepared by heating blood agar. As discussed earlier, the blood agar when heated, it forms brown color, that will be chocolate agar. And last is McConkey agar, which is most commonly used for Enterobacteriaceae members. It contains agar, peptone, sodium chloride, bile salt, lactose, and neutral red. That is a dye, and it is selective and indicator medium. Okay, these were the various Newton mediums used for the culture of bacteria, isolating of the bacteria and identifying the various bacterial colonies. So this was today's class, and in today's class we have dealt with culture media and what is culture media, what are the requirements of a culture media, and we have also classified culture media based on consistency and based on the purpose and the based on the chemical composition. It is a chemically defined or chemically undefined. In the next video, we'll study about the isolation of culture media and the growth of bacteria. And in the following classes, we'll see the measurement of bacterial growth. So this is it with today's class. Thank you for watching.